last part of this section is to talk about mutations, which is probably the most fun part of the section because it raises a lot of questions about what exactly happens when there is an error. Remember, your DNA is 3.2 billion base pairs. It's not super common to have errors, but it's definitely possible. And when that happens, we get a mutation. So a mutation is any change in the sequence of the amount of a cell's DNA. Mutations are caused by mutagens. So these are things that cause changes in our DNA. For example, Rosalind Franklin was using X-ray radiation. Radiation causes changes in the DNA. This would be a mutagen. Other things are chemicals that can cause cancer or any type of mutation in our DNA. There's lots of mutagens in our environment. In some cities, just breathing in the air can be a mutagen. If a mutation occurs in a somatic cell, think back, what is a somatic cell? Is it a sex cell or a body cell? If you think that it's a body cell, you would be correct. So if a mutation occurs in a somatic cell, it affects only the cells of the organism. In other words, it cannot be passed on to the organism's offspring. If the mutation occurs in the gamete, then it will in fact be passed on to the offspring, meaning that we tend to say that it is quote unquote a genetic mutation. However, this is a little bit of a wrong statement because all mutations occur in our genes. So let's talk about some types of mutations. The first type is a point mutation. This mutation only affects one sequence of DNA or gene that codes for a protein. One type of point mutation is substitution. This is a replacement of a single base with a different base. Some things that are caused by substitution is cystic fibrosis. I've talked about this in class before. It's when a person has a large amount of mucus that develops in their lungs regularly, and as a result, they have trouble breathing. Sickle cell anemia is when our blood cells are not donut shape, but rather they're more of a sickle shape or a C or a J shape. As a result, they can't carry oxygen as efficiently, and oftentimes people who have sickle cell anemia have some sort of bleeding disorder. And lastly, color blindness is also an example of substitution. Other types of point mutations would be insertion, deletion, and frameshift. Insertion is the addition of an extra nucleotide into the DNA sequence. Deletion is when one nucleotide is deleted from the DNA sequence. And frameshift is when DNA sequence moves either forward or backward due to an insertion or deletion. So if we take a look at this, we can see what substitution can do. So over here, I have a C which pairs with G. If my DNA sequence were to change and the C were replaced with a T, the result would be instead of arginine, I would have histidine. And that would change my amino acid sequence. Just one amino acid would change. If I look at insertion, I have TAC, GCA, TGG, AAT. If I add a T after my first A, the whole entire sequence is moved over by one base pair. The result is a whole new protein. Not only one of the amino acids remains the same. So I went from methionine, arginine, threonine, leucine to ILE, alanine, tyrosine, and leucine. Okay, this is going to cause a much bigger change in the expression of our traits. Deletion is when one of the base pairs is actually removed. So if I had the sentence, for instance, the fat cat ate the rat, and I delete the first H, I start with something that I can't really translate into anything that makes sense. In other words, the information is hard to read. You can also have a gene or a chromosomal mutation. This affects many sequences of DNA or genes that code for proteins. Deletion can fall into the gene or chromosomal category as well. This is when a piece of a chromosome is removed. Duplication is when a piece of the chromosome or the DNA sequence is repeated more than once. Inversion is when a piece of the chromosome breaks off and is reinserted in reverse. So for instance, if I had HAT, I would reinsert it as TAH. Translocation is when a piece of a chromosome is moved to a different location on the same chromosome. So if we take a look at this, I have A, B, C, D, E, F. My B is completely deleted, and as a result, I get a new chromosome that says A, C, D, E, F. Duplication is when one piece is repeated more than once. So notice my original is A, B, C, D, E, F, and the B gets duplicated. It's in there more than once. Inversion is when I take a piece and I add it on backwards. So notice it should read A, B, C, D, E, F, but as a result, the DE is flipped and it becomes ED, and the BC is flipped and becomes CB. The last is translocation. So I have homologous chromosomes, and pieces of the chromosomes swap locations, but they're on the same chromosome still.